Good morning, everyone. I'm delighted to welcome you all today. Today, we will be exploring the important topic of stability studies. My name is Dr. S.K. Singh, and I hold a PhD in this field. On this first slide, I just want to give you a quick overview of what we'll cover. We'll discuss the fundamentals of stability testing, its significance, and how it impacts our product development. So, let's get started and delve into this fascinating subject together. Today, I want to talk about the purpose of stability studies. Essentially, their main goal is to ensure that a drug product remains safe, effective, and of high quality throughout its entire shelf life. This means we need to verify that the product maintains its intended performance from the moment it's manufactured, until it reaches the end of its approved storage period. One of the key aspects we look at is product consistency under specific environmental conditions. This involves testing how the product behaves when exposed to different temperatures, humidity levels, and light. It's crucial because these factors can influence the stability of both drug substances and drug products. Stability studies are also required for regulatory purposes. They help us get the necessary approvals, determine the correct shelf life, and establish proper storage conditions on the label. This ensures that everyone handling the product knows how to keep it safe and effective. It's important to note that these principles apply to both small molecules and biologics, whether they are drug substances or finished drug products. Understanding these concepts is fundamental as we move forward in ensuring that all drugs we develop and distribute meet the highest quality standards. Let's take a closer look at some key regulatory guidelines and standards that guide our stability testing practices. Starting with ICH guidelines, we have ICH Q1A-R2, which focuses on the stability testing of new drug substances and products. This ensures that we understand how our products behave over time under various conditions. Then there's ICH Q1B, which addresses photostability testing, crucial for understanding how light exposure can affect drug stability and ensuring proper packaging. ICH Q1C deals with stability testing for new dosage forms, helping us adapt testing strategies for different formulations. Moving on, ICH Q1D introduces bracketing and matrixing approaches, which are efficient ways to reduce testing without compromising data integrity. ICH Q1E emphasizes the evaluation of stability data, guiding us on how to interpret and utilize the information effectively. ICH Q1F covers stability testing in different climatic zones, ensuring our products remain stable globally. In addition to ICH, we look at WHO TRS 1010 Annex 10, along with regulatory bodies like the US FDA, EMA, and CDSCO from India. These organizations set the standards we follow to maintain quality and compliance across markets. Currently, our focus areas include ensuring data integrity, performing robust statistical evaluations, adopting risk-based stability protocols, and tailoring stability testing to various global climatic zones. All these elements are interconnected and vital for developing safe, effective, and reliable pharmaceutical products worldwide. Let's start by looking at the different types of stability studies. First, we have real-time stability, which is conducted under the recommended long-term storage conditions. This helps us understand how the product behaves over time, typically over a period of 12 to 36 months, depending on the product classification. It's essential because it provides the real-world data we need for shelf life determination. Next is accelerated stability testing. This method predicts how the product might degrade over a longer period by exposing it to stress conditions, usually at elevated temperatures and humidity levels for about 6 months. It's a quicker way to gather stability data and identify potential issues early. Then we have intermediate stability testing. This comes into play especially when accelerated data shows significant changes. It acts as a bridge between long-term and accelerated studies, helping us confirm stability under moderate stress conditions. Moving on, stress or forced degradation studies are critical for method validation, as outlined by ICH Q1A and Q2, R2 guidelines. These studies involve intentionally stressing the product through thermal, photolytic, oxidative, and hydrolytic conditions to understand degradation pathways and ensure analytical methods are robust. We also need to consider in-use stability, which evaluates how the product holds up after the first opening, especially important for multi-dose products. This helps determine how long the product remains effective once in the consumer's hands. Finally, there's ongoing or annual stability testing. This is mandatory for marketed products, as it ensures batch-to-batch consistency over time and confirms that the product continues to meet quality standards throughout its shelf life. Overall, these different stability studies are all interconnected, providing a comprehensive picture of a product's stability profile and ensuring its safety and efficacy for consumers. Let's talk about the selection of batches first. It's important to choose at least 3 primary batches, which can be either pilot or production scale. These batches should reflect the manufacturing variability, so that includes factors like different API sources and packaging types. This helps ensure our stability data is representative of what will actually be used in the market. Next, we have the Container Closure System, or CCS. Studies must be performed using the final packaging intended for the product. And of course, these studies need to comply with regulatory standards such as USP 661, 671, 1661, or their equivalents. This guarantees that the packaging will maintain the product's stability and integrity over time. Moving on to test parameters, a comprehensive set of tests is required. These include appearance, pH, assay, impurities, dissolution, water content, microbial limits, preservative content, physical parameters, particulate matter, and functionality tests like MDI and DPI if relevant. Covering all these aspects helps us understand the product's stability thoroughly. Finally, sampling frequency is critical for monitoring stability over time. For long-term studies, samples are typically taken at 0, 3, 6, 9, 12, 18, 24, and 36 months. Accelerated studies usually involve sampling at 0, 3, and 6 months, while intermediate studies might be at 0, 3, 6, and 9 months. Proper design of these stability studies ensures we gather meaningful data to support product shelf life and storage conditions. Let's take a closer look at the different climate zones and how they influence storage conditions. Starting with zones I and II, which are classified as temperate and subtropical. For long-term storage, these zones typically maintain temperatures around 25 degrees C with a variation of plus or minus 2 degrees, and relative humidity around 60%, also with a small margin of fluctuation. When we consider accelerated conditions, which simulate more extreme environments, the temperature rises to about 40 degrees C with a similar plus or minus 2 degrees C variation, and humidity increases to approximately 75%. Moving to zone III, this is categorized as hot and dry. Here, the long-term storage conditions are quite specific, around 30 degrees C, again with a plus or minus 2 degrees C margin, but with a much lower relative humidity of about 35%. This reflects the dry heat typical of such environments. In zones IVA and IVB, we see hot and humid conditions. Zone IVA has a long-term temperature of about 30 degrees C with a moderate humidity of around 65%, whereas Zone IVB, which is particularly relevant to India, has the same temperature but a higher humidity of approximately 75%. These distinctions are crucial because they help us tailor storage requirements to each environment. Now, when we talk about refrigerated products, the key point is maintaining a temperature close to 5 degrees C with a plus or minus 3 degrees C variance. This ensures the integrity of temperature-sensitive items. For products that require freezing, the conditions are much colder, around 20 degrees C, with a variation of about plus or minus 5 degrees C. Switching gears to photo stability, as outlined by the ICHQ 1B guidelines, products are exposed to very intense light conditions, over 1.2 million lux hours, and UV energy exceeding 200 watt-hours per square meter. This testing helps us understand how products will hold up under light exposure during storage and handling. Finally, the stability of stored products also depends on the climatic conditions specified by the ICH climatic zones. These conditions are designed to simulate the environmental factors products might face in different regions, ensuring their stability and efficacy throughout their shelf life. Understanding and applying these conditions is fundamental to our overall approach to product quality and safety. Now, let's move on to the key analytical methods we use in our studies, which are validated according to ICH-Q2, R2 guidelines. This ensures our methods are reliable, precise, and suitable for their intended purpose. Next, I want to highlight the importance of establishing clear specifications and acceptance criteria. These define what quality attributes our product must meet to ensure safety and efficacy. Data integrity is crucial, so we adhere to ALCOA+ principles: attributable, legible, contemporaneous, original, accurate, complete, consistent, and enduring. Maintaining data integrity is fundamental to regulatory compliance and scientific credibility. We also develop a comprehensive statistical evaluation plan. This plan guides how we analyze data, interpret results, and demonstrate the robustness of our findings. Change control and deviation management are integral parts of our quality system. They help us track and address any deviations from the protocol, ensuring continuous process improvement and compliance. Our study objectives are clearly defined, aligned with regulatory references, and tailored to meet specific submission requirements. This clarity helps us stay focused and compliant throughout the study. Regarding product details, we include information on the API, formulation, critical quality attributes, and strength. These details are vital for understanding the product's characteristics and ensuring consistency. Batch details such as size and manufacturing process are documented thoroughly. This transparency supports reproducibility and quality assurance. Storage conditions and chamber qualifications are carefully specified to preserve product stability. Proper storage is essential for maintaining product integrity over time. Sampling time points are strategically chosen based on stability profiles to accurately capture how the product behaves during storage. Finally, the stability protocol requirements outline our approach to testing, including testing intervals, conditions, and acceptance criteria, ensuring our products remain stable throughout their shelf life. All these elements come together to form a comprehensive, compliant, and scientifically sound stability study, supporting both regulatory approval and product quality. When it comes to stability chambers, it's crucial that they are properly qualified through IQ, OQ, and PQ processes. This ensures that the chambers meet all necessary specifications and perform consistently over time. Continuous temperature and relative humidity mapping using calibrated sensors is essential to monitor the environment accurately, helping us maintain optimal conditions for our products. Alarm systems play a vital role by providing immediate alerts if any parameters go out of range, and backup power ensures that the chamber remains operational during power outages. Emergency controls are also in place for quick response to any unforeseen issues. Additionally, having standardized SOPs for chamber operation, breakdown management, and contingency handling helps us maintain consistency, safety, and compliance. Overall, thorough qualification and vigilant monitoring are the backbone of reliable stability chambers, safeguarding the quality of our products throughout their life cycle. According to ICH-Q1E guidelines, when we perform trend analysis, we typically use methods like linear regression, mean kinetic temperature, and out-of trend evaluations to assess stability data. These techniques help us understand if the product's stability profile is consistent over time. When variability between batches is low, we can pool data across multiple batches to get a more comprehensive picture. However, any extrapolation beyond the observed data is only acceptable under specific conditions. For instance, if accelerated stability conditions show no significant change in the product, and this is supported by solid scientific justification, then we can consider extrapolating the shelf life or retest period. Speaking of shelf life and retest periods, these are determined primarily based on real-time, long-term stability data. Accelerated data, while useful, only supports predictions. They don't replace actual long-term data for final decisions. Finally, when evaluating data and assigning shelf life, it's critical to rely on real-time data whenever possible. Accelerated data can inform our predictions, but shouldn't be the sole basis for final shelf life or retest period assignments. This approach ensures our product remains safe and effective throughout its intended use. Let's talk about multi-batch stability, which is crucial for justifying shelf life and storage conditions. Ensuring stability across multiple batches helps confirm that our product remains safe and effective throughout its intended shelf life. Next, we rely on stability-indicating validated analytical methods. These methods are essential because they accurately detect any changes or degradation, allowing us to confidently assess stability over time. Full data traceability is also key. From raw data to the final report, and then to CMC submission, maintaining clear records ensures transparency and supports regulatory review processes. We also have the flexibility to use bracketing and matrixing approaches, but only with proper justification, as allowed under Q1D guidelines. This can help streamline testing without compromising data integrity. For biologics, forced degradation studies are particularly important. They must demonstrate that even under stress, the product maintains its integrity and potency, which reassures regulators and stakeholders of its robustness. Overall, these points reflect common regulatory expectations, emphasizing rigorous testing, thorough documentation, and justified testing strategies to ensure product quality and compliance. Let's take a closer look at the key components of our stability protocol. First, we have the stability data sheets, which document all the stability testing results and are essential for tracking how our product performs over time. Then, there are trend reports, which analyze this data to identify any patterns or shifts that could impact product quality. Annual product reviews, or APRs and PQRs, are also critical. They provide a comprehensive review of stability data over the year, ensuring ongoing compliance and product integrity. Change control assessments are another vital aspect. They evaluate any modifications to the process or formulation to confirm they don't negatively affect stability. Regulatory submissions, specifically Module 3 in the Stability section, require detailed documentation of our stability data to meet regulatory standards. Lastly, the stability study documentation encompasses all records related to our stability studies, serving as the backbone for demonstrating product quality throughout its shelf life. Altogether, these elements form a robust framework that supports the overall integrity and compliance of our product, aligning with our goal to deliver safe, effective, and high-quality medicines. Now, let's talk about some of the key challenges and best practices in this area. First, chamber failures and data loss can really impact our processes, so it's crucial to implement redundant monitoring systems to catch issues early and ensure data integrity. Moving on, managing out-of-specification or out-of-trend results should be aligned with industry guidelines like ICH Q14 and Q2, R2, which provide a solid framework for quality control and reporting. Next, adopting a quality-by-design approach helps us better understand degradation pathways. By doing this, we can proactively identify potential stability issues before they become critical. To support this, utilizing risk-based stability assessments, such as temperature cycling and transportation studies, allows us to simulate real-world conditions and predict product performance more accurately. Finally, regular review through product stability review meetings keeps everyone aligned and ensures ongoing oversight. These reviews are essential for catching trends early and implementing continuous improvements. Overall, integrating these practices helps us maintain high-quality standards and stay ahead of potential stability challenges. Stability studies are really the backbone of ensuring our product maintains its quality over time. They help us understand how the product behaves under different conditions and ensure it stays safe and effective until its expiration date. Compliance with international guidelines like ICH, WHO, US FDA, EMA, and CDSCO is absolutely mandatory if we want our product to be accepted globally. These organizations set the standards that we must follow to meet regulatory requirements across different markets. To achieve this, having a robust design for our stability protocols is essential, along with validated methods that guarantee our results are accurate and reliable. Data integrity is also a key pillar, making sure our data is complete, consistent, and trustworthy throughout the process. In conclusion, focusing on these core elements, stability studies, compliance, robust design, validated methods, and data integrity, ensures we deliver high-quality products that meet global standards and ultimately benefit patients worldwide. For further support, please don't hesitate to reach out to us. We're here to help and provide any additional information you might need. Feel free to contact us at your convenience. And, of course, thank you very much for your time and attention today. We truly appreciate your interest and engagement throughout this presentation.